Welcome to this virtual lesson from SPARQ. Our topic today comes from Unit 2 of the Biology syllabus, Homeostasis. As a quick warm-up, sketch a diagram to represent the structure of cell membranes. See if you can include and label phospholipids, cholesterol, glycoproteins, protein channels, and the words hydrophobic and hydrophilic. My name is Charmaine Keel and I am the teacher and the head of department at SPARQ. SPARQ is located at the Translational Research Institute in Woolloongabba. The program is supported by the Queensland Government Department of Education and the University of Queensland. It is our mission to inspire and engage students in STEM with a particular focus on biomedical research. How did you go with your warm-up? Did your diagram look something like this? Take a moment to pause the video and have a look at the diagram. The diagram is clearly labelled from A through I. See if you can match A through I with words from the word bank on the right of the screen. Pause the video and write your answers in your notebook. You can check your answers with this list on the right of the screen. If you got more than 8, you did really well. Congratulations. This virtual lesson will cover content from the beginning of Unit 2. In Unit 1, you should have learnt about cells, enzymes, energy transformations that occur within cells, how cells differentiate to become specialised, and explored some systems including gas exchange, transport, nutrient exchange and waste products. By the end of the lesson, you should be able to define homeostasis. We will have explored the stimulus response model, including the roles of receptors, effectors, and feedback cycles. We will also look closely at the feedback cycle for glucose regulation and explore some issues around diabetes. Homeostasis literally means same state. So it refers to the relatively constant physiological state inside living things. Organisms maintain this constant internal state despite many changes in the environment around them. Examples of variables that are maintained are things like temperature, pH, blood sugar levels, and water. This diagram represents a simple feedback cycle for maintaining homeostasis. Down the bottom, you can see variables. When in balance, they're considered to be in homeostasis. When imbalanced, you can see if we move the arrow up, the changes are detected by receptors. Those receptors send information to the control centres at the top of the diagram. The control centres then forward output information or a way to respond to the change to effectors. The effectors then work to bring the organism back into homeostasis, which balances the beam. I'm going to give you some definitions for some of the terms mentioned in the previous slide. The role of a receptor is to detect any changes in the environment and send a message to the control centre. There are 5 types of receptors that the syllabus asks you to be familiar with. We're going to look at 4 of them in more detail. The one we're not going to look at is the nociceptor. A nociceptor is a type of pain receptor. The function of the control centre is to receive messages from the receptors and coordinate a response. They then send an output message to effectors. In humans, our main control centre is our brain, but our spinal cord has some ability to send our output messages back to effectors. Effectors' job is to respond to the output from the control centre, thus bringing us back to homeostasis. We have 2 main types of effectors. They are muscles and glands. Receptors respond to stimuli by producing an electrical impulse. In this way, they act as transducers, converting energy from a stimulus into the electrochemical impulse. They do this because the stimulus opens or closes protein channels which allow ions to flow in and out of the cells. This flow of ions causes minute changes in the potential energies along the cell membrane. The diagram shows 4 different types of receptors. The first on the left are mechanoreceptors. These are the simplest. Pressure, bending or stretching simply opens the ion channel and allows, for example, sodium to travel through. The second type of receptor is a thermoreceptor. You can see that warmth or change of temperature triggers an enzyme which then opens or closes the ion channel. In the third, a chemical binds to a receptor on the surface of the cell membrane. This initiates a signal or chemical reaction cascade which eventually will open or close the ion channel. Finally, a photoreceptor works similarly, except this time it's light that alters the membrane protein, initiating the cascade which opens or closes the protein channel. Remember that homeostasis is about maintaining that steady state inside an organism. One of the main ways to do it is through a negative feedback cycle. In a negative feedback cycle, receptors detect departures from the normal and act to restore it back to the set point. This dampens any variations caused by the environmental changes and has a stabilising effect on systems. Let's look at an example. On the right of the screen is the negative feedback cycle for temperature. We know that generally our internal body temperature remains between 36 to 38 degrees. If the temperature rises above normal, that is detected by your thermoreceptors and an impulse is sent to the brain. The brain responds by sending signals to the sweat glands and the blood vessels. At the sweat glands, sweat is released, which evaporates, cooling the body down. At the blood vessels, these dilate, which cause our skin to look red, and again we radiate heat out, which cools the body down, which drops our body temperature back towards homeostasis. If you cool too much, the bottom cycle might happen. So the temperature falls below normal. Again, the temperature receptors detect this change and send a signal to the brain. The brain can then send a signal to effectors like the muscles, which cause shivering. That shivering generates heat, which raises the body temperature back to normal. So you can see you have, if we graphed this, you would have an increase in temperature here, which raises the body temperature. As we sweat or vasodilation occurs, the body temperature decreases. If it decreases too much, the hypothalamus detects that change and responds by shivering or by vasoconstriction, which causes the temperature to rise. This fluctuating graph is classic for negative feedback. Another type of feedback cycle is a positive feedback cycle. It has a very different function to negative feedback. Negative feedback is about maintaining homeostasis, whereas positive feedback is about pushing the body beyond the normal to achieve a particular result or outcome. The response stops when the result is achieved. An example is childbirth. During childbirth, the head of the baby pushes against the cervix. That stimulus targets receptors, which sends a signal to the brain. In the brain, it stimulates the pituitary gland to release a hormone called oxytocin. Oxytocin travels through the blood to the uterus. The muscles of the uterus are stimulated by the presence of oxytocin to contract, thus delivering the baby. Once the baby is delivered, this cycle will stop as the stimulus of the head pushing against the cervix is no longer there. I have a question for you. If a negative feedback gives that classic fluctuating graph, what do you think a graph of positive feedback might look at? Pause the video and take a moment to sketch your graph. Let's check how you've gone with the key points so far. Again, pause the video and match the key terms to the definitions. You can simply write in your book, for example, if you think the definition of a chemoreceptor is C, then you would write 1C. Pause the video and have a go at those 8 key terms now. Let's take a moment to check a couple of those answers. Key term 3 was a mechanoreceptor. A mechanoreceptor is a receptor that detects distortion as caused by touch or pressure. So F is the most appropriate response. Negative feedback, key term 4, is a mechanism where the output acts to oppose changes, which stabilises the system. So definition A was the most appropriate. A nociceptor is a special type of receptor stimulated by potentially damaging stimuli. So C was the best definition. Give yourself a mark out of 8 for how well you did on that quiz. The remainder of this virtual lesson will focus on glucose regulation and what happens when it goes wrong. Glucose is a simple sugar, a monosaccharide, based on 6 carbons. It's hydrophilic due to the large amounts of oxygen and hydroxyl groups. Glucose is utilised within your cells for cellular respiration, where it's converted into ATP, a form of energy that your cell uses easily. Glucose can cross the cell membrane using facilitated diffusion. Usually, the outside of the cell will have a higher glucose concentration, where the inside of the cell will have a lower concentration. So it wants to move inside, and it will go through special protein channels to let it in. Glucose levels in your blood is controlled through a negative feedback cycle. After eating, your blood sugar or glucose levels will increase. The pancreas detects these changes and releases a hormone called insulin. Insulin travels through the blood to the liver, where glucose is then converted into a chemical called glycogen and stored in the liver. This reduces your blood sugar level back to normal. On the opposite spectrum, during a fast or if you've been a long time between meals, your blood sugar levels will decrease. Again, the pancreas detects these changes and it does 2 things. It stops insulin production and actually releases a different hormone. This one is called glucagon. Glucagon travels through the blood to the liver, and when there, it converts the stored glycogen back to glucose, thus raising your blood sugar levels back towards normal. Take a moment and have a good look at this diagram. Make sure you understand it before you move on. A key component of glucose regulation is the action of insulin to reduce the level of glucose in the blood. Let's look more closely at the mechanisms in cells that are responsible for this. It was mentioned before that glucose tends to travel via facilitated diffusion through protein channels. Some of these channels act independently of insulin and some only function in the presence of insulin. GLUT4 is an example of an insulin-dependent pathway. Diagram A illustrates these structures near the cell surface. You can see the insulin specific chemoreceptor, here, located within the membrane. Sitting just inside the cell, within the cytoplasm, are secretory vesicles. These secretory vesicles consist of a layer of cell membrane and a couple of transport proteins. Because these transport proteins are not located in the actual cell membrane, glucose is not able to enter the cell, and the concentration of glucose outside of the cell remains high. When insulin binds to the chemoreceptor, a cascade of chemical reactions begin. That's shown here on the diagram. The end result of these reactions is the merging of the vesicle to the cell membrane. As the vesicle merges with the cell membrane, the transport proteins become available to move glucose inside of the cell along the concentration gradient. As glucose moves inside the cell, the amount of glucose outside of the cell reduces, therefore lowering your blood sugar levels. The graph illustrates relationships between time of day, blood glucose levels, and insulin. There's a lot going on here, so let's focus at one part at a time. I'm going to model analysis, a simple analysis of this graph. The solid red line represents blood glucose from starch-rich foods. It is clear that glucose levels fluctuate throughout the day. The level starts at approximately 4.3 millimoles per litre, and there are 3 high peaks throughout the day that seem to correlate with meal times. At the end of the day, the level slowly drops before stabilising between 4 and 5 millimoles per litre. The blue line represents insulin levels. After each insulin peak, the level of blood glucose drops. The insulin also drops at this time. This graph also includes information regarding other types of sugars that were included at lunchtime. It's often said that consuming foods with a large amount of simple sugars like sucrose and fructose can lead to a sugar high and then a sugar low. Here's a question for you. Does the information in the graph support this statement? Pause the video and write a brief response to this question. A high-level response would include specific evidence from the graph, so state numbers and make comparisons. The regulation of blood sugar doesn't always work the way it's supposed to. Diabetes is a chronic metabolic disease which is characterised by high levels of blood sugar. Over time, these high levels of sugar in the blood can cause serious damage to the heart, kidneys, blood vessels, nerves, and eyes. There are 2 types. There's juvenile or insulin-dependent diabetes, type 1, or type 2, which is adult diabetes. For people living with diabetes, they need to have access to insulin. The manual intake of insulin, whether through injections or tablets, is critical for their survival and for maintaining a fairly regular blood sugar level. The World Health Organization is reporting that 422 million people currently have diabetes across the globe, and approximately 1.6 million people die annually from complications. In November 2019, the International Diabetes Federation released this data about the number of adults with diabetes worldwide. Pause the video here, and I'd like you to write down 5 observations you make on viewing this data. SPARQ is located within the Translational Research Institute. Several research groups within TRI are studying diabetes. One group is led by Professor Josephine Forbes from MARTA Research. This group has been studying AGEs. AGEs are advanced glycation end products. They are the result of chemical and physical processing of food and are ingested when we eat highly processed foods. The group's results have shown that AGEs can increase the likelihood of developing diabetes. They also increase in production within the body if you have diabetes. And finally, AGEs contribute to the complications of diabetes. Professor Forbes's group focused on heart and kidney disease in terms of complications. Studies involving animal models suggest that lowering the intake of AGEs in their diet reduces the onset of diabetes. Also, medications that slow the formation of AGEs reduce the onset of diabetes and its complications. The team is now focusing on translating these animal studies into human trials in the clinic. That brings us to the end of our lesson. Take a moment, pause the video and go back through your notes. Make sure you have responses to each of these and that you feel confident in responding to them. Thank you so much for watching. If you have any questions or feedback, you can contact us at sparked@uq.edu.au. Thank you and stay safe.